Welcome to my channel, Correct Biology. In this video, I am going to teach you about test cross and back cross. Here you must have learned that test cross, it is being performed to know unknown genotype. While the back cross, do you have the answer? Yes. Why actually we perform the back cross? No. Be with me and learn clearly what is test cross and what is back cross. Now here I just want to tell you, test cross, it was being developed by G.J. Mendel. Hopefully you know G.J. Mendel. He is well known as father of genetics. If you remember, Mendel, he carried out monohybrid cross. We have already discussed monohybrid cross, dihybrid cross, okay, using the pea plant. And here, if you remember, I have told you, he actually started his work using true breeding varieties. In today's sense, these true breeding varieties, they are homozygous. They are going to have same type of allele. Okay, when you are going to have tall plant, and if both the alleles they are same, then this condition is known as homozygous. Now try to see here. If you see in the case of monohybrid cross, when Mendel, okay, when let's suppose you are going to select tall plant, okay, now there can be two genotypes. One, here it can be capital T, another it can be capital T, small t. Now here if you see in the case of dwarf phenotype, okay, so here if you see, if the plant is dwarf, okay, so what you are going to see, the genotype is going to be 1, small t. So this we have learned when we have learned about the monohybrid cross. Now try to see, let's suppose you are going to have a tall plant, okay. So just by looking the phenotype, you are not going to tell what is going to be the genotype, okay. Okay, so you have to devise some test. So at that time, okay, Mendel, okay, he also devised one test. By performing that test, you can easily tell whether the plant it is homozygous or heterozygous. What was the test? Okay, now try to see here. Let us suppose you are going to have this one homozygous, okay, true breeding varieties, okay, and if you are going to cross it with homozygous recessive, okay. So Mendel, what he did, he crossed the tall plant with homozygous recessive. So here you are going to see the formation of one type of gamete. Here also you are going to see formation of one type of gamete, okay. They are going to fuse, and here in the F1 generation, what you are going to see, the genotype capital T, small t. So this is F1 generation individual, and here what you are going to see, 100% tall. So all the F1 progeny, they are going to be tall. Now take another situation. Let's suppose you are going to have this heterozygous, okay, genotype is heterozygous, capital T, small t, and you cross it with homozygous recessive, okay. Here you are going to see the formation of two type of gamete, capital T and small t. Here you are going to see the formation of one type of gamete, and here if you solve this by Punnett square, okay, so here what we are going to see, capital T, small t, and here small t. And if you solve this, capital T, small t, small t, small t. So this is going to be tall, okay, and this is going to be dwarf. So here what we are going to see, 50% tall and 50% dwarf, okay. Now try to see, if you are going to cross, okay, with homozygous recessive, okay, if you are going to cross unknown genotype, okay, with homozygous recessive, and if you are going to get all the F1 progeny, they are going to show the dominant phenotype. If all the F1 progeny they are showing the dominant phenotype, that is 100% tall, means the unknown genotype, it is homozygous, it is true breeding variety. Now same, if you see here, if you are, let's suppose this is unknown, okay, and if you are going to cross it with homozygous recessive, and if you are getting 50% tall and 50% dwarf, then you can say this unknown genotype is heterozygous, okay. Now by performing the test cross, you can easily tell whether the plant it is homozygous or heterozygous. How we actually perform the test cross? We cross the unknown genotype with homozygous recessive, okay. So it's quite simple, okay. You cross, so let's suppose there is unknown genotype, okay, and you want to know what is the genotype, you cross it with homozygous recessive, okay. If you are going to get all, okay, F1 generation, they are going to show you dominant phenotype, okay, then you can say the unknown genotype is homozygous, okay. And if you are going to have the result that you are going to have 50% progeny are showing the dominant phenotype and 50% progeny they are showing the recessive phenotype, okay. In that situation, the unknown genotype is heterozygous. Hopefully you have understood what we do in test cross. In test cross, we cross the organism with homozygous recessive individual, and purpose of the test cross is to know the unknown genotype. Now we are going to discuss the back cross. Now in back cross, let's suppose we are going to have one organism, let's take the mouse, okay, and we are going to cross it with either of the parent. We cross it with either of the parent. Yes, next generation you are going to see progeny, okay. In the next generation you are going to see progeny, okay. Now this one, it can have any genotype, okay. Here also it can have any genotype, okay. This can also have any genotype, okay. Now this back cross, it is not going to tell you the unknown genotype. If you want to know unknown genotype, you have to perform the test cross, okay. But here if you want to know why actually we are going to perform back cross, try to see here. Let's suppose I will make you understand. Let's suppose you have created a mouse, okay, by using some transgenic technology, okay. You have created a mouse, okay, by transgenesis, okay, and you have introduced X gene, okay. So you introduce X gene within the mouse, and this X gene is foreign, okay. Now you are going to have a new variety of mouse, okay, and in this mouse you are going to have X gene, Now during the time of transgenesis, you are going to follow certain procedures, okay, and during the time what might happen, some of the essential genes, like let's suppose we take Y, Z, okay, they might be lost. So this has become, this mouse, let's suppose you test it, it is X positive, but now it has become Y minus, Z minus. Now how we are going to retain back these Y and Z genes? It is by back cross. So Y and Z genes, okay, they are present within the parents. So here in the back cross, okay, what we do, we cross with either of the parent, okay, we cross it with either of the parent. What is the purpose? Okay, to restore back the genes which were being lost earlier. So here you will keep doing the cross until it is X positive, Y positive, Z positive. Okay. So this is the purpose of performing back cross. Hopefully you have understood what is test cross. Okay, test cross. In test cross, we cross with homozygous recessive parents. Okay, we cross with homozygous recessive, okay, individuals, okay, not parents, okay, so that we can find the unknown genotype. Okay, so keep in mind, in test cross we cross with homozygous recessive individuals, not parents, to know the unknown genotypes. In back cross, okay, we cross with either of the parent, and here I have told you what is the purpose, to restore genes. Okay, so hopefully you have understood what is test cross, what is back cross. Please do like, share the video. Okay, if you have any specific comment, please do comment, and thank you if you have subscribed my channel Correct Biology. Correct Biology. Thank you.